Okay, I'm going to cover a lot in this presentation. Normally I'm sort of putting into what I would do in 4 days into 45 minutes, so hang on for the ride. There's going to be a lot of information come at you. I'll be around for the 3 days, or we do more in-depth seminars where we cover all of these different parts, like a full day. So I'm just going to do a very quick general overview. As we were talking a lot about humus, we've registered humus technology quite a few years ago, and since then the word humus is just coming up more and more and more, and it looks like it's going to be humus. I was talking to Jeff Baldock the other day that when and if we ever get to trading soil carbon, that we'll actually be measuring how much humus we've built in the soil. So it's a pretty important word. It's a word that has been around a long time. I think we know it, but it's certainly going to come to the fore a lot more. So this is just building soils, a photo. Okay, so that's me and Bill at 22 when we were courting. Aren't we a handsome couple? So today I'm going to cover an overview of humus compost production, the quality parameters, which I think a lot of people get hooked up with because compost ain't compost. And not that, you know, regular compost does its things, but we've chosen to take our compost to a very high standard, more like a very high quality fertilizer. I put it out to scientists everywhere I speak or everyone I meet. I put it out to them that it's the only fertilizer I know that actually benefits the 3 aspects of the soil. And I'm waiting to have a good debate with someone that they say there's something better. No one's done it yet, so I welcome anyone because I still haven't found anything that will benefit those 3 aspects of the soil as much as the humus compost does. We're going to cover some trial results. I think it's important that you see some trial results and some scientific measurements that have been done because everyone says, how is it proven or is it proven? Not that I try and prove myself to science too much because it is flawed as well. But I've just got to say, and it just came back to me then, that when we started in 2002, I really thought if I can prove myself to the CSIRO and the DPI, then I'll have achieved it. Well, about 6 weeks into running Wild Ad Living Soils, guess what? I realised that 10 years ago was not going to happen. And I realised that I just had to forge ahead despite what they thought. Because back then, GRDC, CSIRO, DPI didn't even believe there was life in the soil. So this was just silly Rhonda Daly off on her little track of, you know, something stupid. But now today, it's accepted and lots of money's been put into researching. Gupta does a lot of research in Adelaide. And we're also searching for it from the carbon perspective as well, as a potential that soils are the 2nd largest sink to sequester carbon. Our oceans are the largest, of which are getting acidified from taking in too much carbon because we can't stop that. But we do know that our soils are the 2nd largest sink of which to sequester carbon. And all us farmers, or is that good grammar? I'm not sure, but we can all help that and benefit. So at the moment, I say there's a benefit from building soil carbon just from production and profitability and sustainability in your own health. But I think down the track we will see more people come on board this if they're going to be paid, you know, $25 a hectare every year. So I think it's important we still keep going there. We've got the Kylie speaking on Friday about trading soil carbon. So it's important to think about it. But where we came from and where we started Wild Ad Living Soils was to put life back into the land because global warming really wasn't spoken about at that stage. What is soil? This is really not what is soil, it's real more the aspects of the soil, and we've got to look at this balanced view of what soils are. You know, they have air, water, minerals, clay, silt, sand content, and of course bugs living in it. But the soil, when you speak about the aspects of the soil, we talk about the physical, the chemical and the biological aspects. Now we need to get all of these 3 working together. I want to go back to that because what happens in conventional agriculture is we've really only focused on the minerals and a little bit of the more on theory. The more minerals we put on, the better it is. But this year, you know, 10th year into business, is I've found conventional farmers coming in and what they put on in their minerals are actually not delivering the results that they would think that they would get. So, but in humus or biological agriculture, we look at these 3 aspects of the soil. I'm going to go back to this slide. This is what Edwin says, the 9 critical factors. So we look at each 3, but he has 3 within each 3. Now remember, someone said if you just use one product it doesn't work, and you try another one it doesn't work, and you try another one it doesn't work. Well, what Edwin says, if 5 out of 9 of these parameters are not in alignment, no one product will work. So you might just put on one biological or one lot of microbes and say, well, that didn't do anything. But if 5 out of 9 are in balance, or the measure they should be, any one product will work. So basically you're looking at the health of your soil to determine how much remediation it needs. You know, where are you with your soils? And that's where I think we pride ourselves. I was thinking the other day, you know, chemical agriculture, you go in and buy your Roundup, you go and buy your fertilizer and whatever. Every customer is just treated like personally, because your soils are so personalised between you. So we can identify where you're up to, you know, 9 or 5 or 6 or 3 as to what you need to do to get your biggest bang for your bucks. And so, as I say, there's that snake oil thing. Oh, it's snake oil, it didn't work. No, I don't think it was snake oil. I think your soils were so bad that it didn't have a chance. So just remember that fact as you're going forward. So what is compost? It's organic matter that is broken down. And this is most compost. So what you've got, if you've got a leaf, it breaks down into cells, and then all the glues and resins and the protoplasm inside the cells. So you end up with a lot of stuff. The difference with humus compost is when we put in an inoculum, we put in more microbes, and we build it up into a long chain polymer of carbon. Hugh was talking about the humus flywheel. What we're saying is it's a humus polymer. This is it under the electron microscanner, the accordion shape of the humus when it's pictured at really high resolution. So what humus compost is, it's organic matter that's been broken down by microbes, but then another set of microbes come in and eat it again, so it goes through the body of a microbe, and they put another carbon onto it. And so that you end up with this big carbon chain, basically. So this is the difference if you just break it down and you don't build it up. But we go to the effort, and that little bit of extra effort, we'll talk about, of building it up. Now, when we were in Corona, Switzerland 2 years ago, we went for a walk in the deciduous forest. It was pretty dark, as you can probably see up there, and sort of a bit of eerie and whatever. And we came across, or Bill saw it, we came across this washaway. Now, we could see humus build up over probably hundreds and thousands of years, but here was the parent material. So it's really not that very good soil, but all of this deciduous, of course, forest, the leaves are broken down naturally. Now, who of us can afford to take our property out of production, put deciduous trees on it, let them rot down, in 100 years we might have some humus? We can't. But what we're doing with making the humus compost is we are just accelerating what nature does naturally and building this humus layer for ourselves. So here you've got the value and the extra effort you go to. You have waste disposal compost, you have good or regular compost, but you can go to that extra effort and you can make humus or humified. Someone said the other day that all compost should have an adjective in front of it. I thought it was really good, really, because you have industry compost, regular compost, humified compost, and there's another one starting with Is Compost that I use. But when we started Wild Living Soils, we were not in waste disposal. So we didn't think, we're getting paid to take all of this waste, now let's do something with it. We went out there to produce the highest quality fertiliser to restore our soils that we could. So we've never come down here into this level. We've always wanted to pitch for the top. And a little bit of extra effort to do that, it's 1,000-fold. I can't tell you how much benefit it is. So the value of humus compost comes from its ability to restore or enhance the biological activity in the soil, allowing nutrients to cycle and all the functions of the soil to get back to normal. Because really what we've done with conventional agriculture, and as a 4th generation farmer I've seen it, we've actually just deadened our soils. You know, Christine Jones has got a lovely saying, we've turned a fluffy chocolate cake into terracotta tiles. So now we've got to go back the other way, which explains it well. So we started composting in April 2006, not very successfully. I had an old turner and had windrows laid out, and I would get it to about 3 weeks, and we'd do one turn with this old turner, not the one Cam got because that's a good one, and I'd turn it into balls. Literally, the whole windrow would be balls. So I'd ring Edwin up in America and say, what have I done wrong? What have I done wrong? And of course it's very hard to tell advice from, 12 hours by the plane away. And by the time he got there in September, I had probably nearly a pile as big as this room of balls. And it would just, every time it would happen. And really, you know all it was? Well, other than the design of the turner, but my water application was wrong, incorrect. My droplets were too big, and I was putting it on the compost before it was in the air. So it was simple as that. So, yeah, just because if you go into composting and make some mistakes, don't think we've all done it. We've all had those problems. So the management metrics and processes phase of the compost, this is not a composting presentation. If you want to learn more, then you've got to come to a composting 101 day. The management, now let's go through the phases. There's 4 phases in producing humus compost. The first one is synchronization, which Hugh started speaking about. It's when you break down your carbon source, you start preparing it, so it will break down at the same time as your nitrogen source. So then you have breakdown, which is week 3 to week 6, and humus formation or build-up is week 6, or we start about week 4 to week 8, and then stabilisation. So when you get a compost, it shouldn't be hot, you shouldn't see what it was made from, and it shouldn't be smelly. So the Compost Australia got all tests from all over Australia to see the maturity of it. Our compost was the only one that didn't reheat. All of the others, as soon as they wet them, they started to come back to life again. Does anyone think that we've got a lot of Pac-Man in that compost that just go along and chomp in on the carbon? Chomp chomp? Does anyone know how it breaks down? What it is, it's the moisture, and cells burst open. So it's actually not a lot of Pac-Man going chomp through the straw. You burst the cells open and then the microbes eat those cells. So it is critical to get the recipe correct, 25 to 30 to 1 carbon to nitrogen. Aeration, we are making an anaerobic, so that's why we measure the CO2, which you'll see at the site this afternoon. Anything over 10, it gets turned because we want to keep it aerobic. If it goes anaerobic, you'll produce what's called sulfides, and sulfides are detrimental to plant growth. So that's why some people say, well, I used compost and it didn't work very well, because it could have been full of sulfides. And the other thing is moisture management, because if you don't get your moisture right and you're not bursting cells open, it will end up just rotting away. And temperature. Once again, we don't want to burn it and we don't want it too cool because we've got to kill E. coli and salmonella and weed seeds. So when you spread it, you don't want to get a whole lot of weeds from someone else's property. So these are the 4 stages: synchronization, breakdown, build up and stabilization. And this is done in a 10-week phase. So not like the old world forest where it takes 200 years. Although I suppose every year it would be breaking down that layer. This is a photo of our site, so you'll see new compost windrows here that are still quite hot. The straw laid out, compost here that's probably just been turned, and we cover from week 3. You build 2 windrows together. They shrink by 50%. You're not losing weight, you're just losing volume as it starts to break down, and then we join them together at week 3. That's when we start putting our covers on. Edwin would like us to put the covers on from day 1, but our windrows are very big because we've got the 170 turner and they're quite heavy, and so we come in at week 3 and start covering the compost. And I love Hugh's comment about the skin. It's very good. I was actually visualising everyone sitting here without, well, I won't say without their clothes, but without any skin on, because it would be probably a gruesome sight. Yes, we put the straw out, we wet it up a few times, we add all the different feedstocks, and I'm not going into recipe building, but there would be, we could start with straw, we could have some old loosened silage, cardboard, paper. So anything that's high carbon, wood chips, green waste. And then you add your nitrogen source to it. So that can be manures or green manures, and they're probably the best sources of nitrogen. And then you turn it in dry, both ways dry, before you add any moisture. Because what you want in the finished row, when you've finished building it, you want every particle touching every other particle. So if you pick up a piece of straw, you want to see pieces of clay on it, you want to see pieces of manure on it. So you don't have big lumps of clay and big lumps of manure. Everything's touching everything else. And then once you've added all your feedstocks, you bring up the moisture level to 45%. So that normally, see there's little Landini still. I love that tractor, I don't like the big new one now. You bring this up to moisture, and then that's really when I say day one, everything starts cranking. So the next day you'll come out, temperature will be up, CO2 will be up, and you'll have to turn and it'll need moisture. The reason to add clay is the foundation of the humus crumb structure. I was talking to Jeff Baldock the other day, and he was saying that all of this measurement they've done of the carbon in Australian soils, where there's a higher clay content in your soils, they're able to build humus quicker than in our sandy soils. So this is where, we ensure that farmers know their soils. Have you got high clay content, so you know what's going on in your soils itself. So to add the clay to the compost, it's for moisture management as well, odour control, because you don't want to stink out your neighbours. You capture more nutrients, because remember clay hangs onto your positive minerals, and it reduces shrinkage, and as I said, it's the foundation of that humus crumb structure. So week 1 to 3 this is the big windrow, which by week 3 will shrink. So as I said, it shrinks in volume, not weight. And this is where the most CO2 will be emitted. Nearly every day you'll spike what's called 20 plus. And we won't go into the oxygen, carbon dioxide in the atmosphere, but 20 plus is the most you can go. If it's over 10, we'll turn it. And if you get a lot of this white fungi in it, you want to turn. It means it's going anaerobic and it's too hot. So I don't know if anyone's ever bought any compost where it's actually still steaming and it's got this white friendly fungi through it or not, but it's certainly not finished compost. So this is the breakdown phase, when cells are bursting open and microbes are chomping into it. So each day we measure temperature with a probe, and we'll practice all this afternoon. Moisture, it's probably the hardest thing you learn, because there's no moisture monitor that will actually work. You need to do it with your fingers, and you just get used to it. What percentage is that moisture? Or the question you're actually asking is, does it need water or not? And if it needs moisture, then how much moisture does it need? And as I said, the carbon CO2 reading will range from 0 to 21. So here's Ross Marsh, testing the carbon dioxide. So at the beginning, your temperatures will be high, your CO2 will be spiking every day. Can anyone tell me why CO2 would be spiking every day at the beginning? I want you to start thinking about this. So when you go out there, you'll see a windrow that's new. So what do the microbes give off? CO2. And what happens when something's decomposing? It gives off CO2. So you're going to have the highest readings of carbon dioxide at the beginning, always. So hence that means you're going to be turning the windrow more at the beginning of the process than at the end of the process. Week 4 to week 8, from week 3, when there's no smell of ammonia in the compost, we add what's called a humifier. We bring that in from America. It's a certain trophic level of microbes that are the ones that build it up. Naturally in the feedstocks you use, you've got the native microbiology that will break it down, but we don't have the ones that will build it up. So these have to be added to the compost, about a litre a turn, and we put that in about 12 times. So this is a stable compost. It's been converted from all of that carbon and nitrogen and all that mixed together into this stable humus compost ready for application. Now when we apply it, and I'm not going to get deep into how much or what and for what soils, but really the smattering that we put on, it's got to be seen as a catalyst. You know, there's not enough there really to do what it does. And what it does is just ignites the whole soil system for it to start functioning correctly. So, because, you know, just been at a compost day and they're going to put on 37 tonnes to the hectare of a regular compost. So Rhonda's sitting there shuddering all day because they think that's what will build soil carbon. And it's not. It's actually the plants that are going to build your soil carbon. It's just getting everything to work. All the minerals in balance, enough humus in there, enough microbiology, the roots and the plant are really what's going to make everything work. So this high carbon compost to make high carbon soils is a bit flawed. So the difference between regular and humus compost, you really go about halfway. You break it down, but you don't get to the growing up and the build-up stage. So this is the little bit extra that we go to. The quality of the compost, the parameters that we go to, we mentioned an absence of sulfides, so that's sulfur attached to CO2 rather than oxygen. Nitrites, so we have nitrates in it, but we don't want nitrites and heavy metals. It's aerobically produced. There's an absence of pathogens, E. coli and Salmonella. There's a high rate of humus. The N fixed into stable humus and microbial biomass. There's an abundant and diverse range of the 5 functional groups of microbiology, the stability and maturity index, and there's a germination and a vigour percentage as well. So all of these tests will say, you know, we haven't really talked much about minerals there, have we? You know, we're actually talking about how is it produced and what's the humus fraction, even though there are minerals in that. This was assessing Professor Petra Marshner, who I must apologise, who's meant to be here Friday. She's had a lecturer get critically ill and she can't leave the university. So Hugh Lovell and John Crawford are going to pick up her presentations. But they tested 4 different composts at the Adelaide Uni for me. And the Wildad compost had high concentrations of total and available P. It was the correct pH, which you was talking about, from 7.3 to 8.1. Ours was 7.6 in calcium chloride. It had low organic matter, but the humus content was high. It germinated seedlings, had longer roots, and it was a finer texture. So the effects of the Wildad humus compost on the soil properties compared to 4 other composts, it increased the pH of the soil, it decreased the EC, the electrical conductivity, it increased the P, and it increased the concentration of particulate organic matter, and it increased root growth. I'm not going to go into the fractions of carbon. Does everyone know their fractions of carbon? Just yes or no? Because I think it's something we really want to go into at some stage, that you understand your 3 pools of carbon. As I say, I'm just chomping this right down, but it is important. Because I say if you're a jeweller and you didn't know how to make jewellery or fix it, you're going to be unsuccessful. If you're a farmer and you don't know your soil, you're going to be not very successful. So I think the biggest thing on your action list is to get to know your soils intimately. And in the paddock, we'll smell it, we'll feel it, we'll tread on it, and we'll have a look at it. But, you know, this is what I like my customers to know, is what's happening in their soils. This was another test done at Adelaide by Petra. It was just testing phosphorus in the soil, pH and phosphorus, and there was 3. There was Jeffreys, Amro and FMs, and ours had 365 mgs to kgs of available phosphorus, and the pH was good. So you're getting a lot of phosphorus in there. This was another one that showed humification. So it's basically, it boiled down, she said, and it's written back to front because she's European, the various absorbance reading indicate good humification of the compost property. So a lot of people will say, well, how do you measure it? Well, it is measurable. And I think with technology, well, I don't think with technology, I know with technology that we're able to understand our carbon, our minerals, and what humification much more, because we're getting so sophisticated. in all our testing and our mechanisms of testing it. So humus compost is more than just NPK. So like anyone, you can present manure and it will have an NPK value. But is it going to do for the soil? Is it going to improve your humus content? Is it going to make the beneficial microbes and your free workers come back? And are the minerals available? This is a Hugh Lovell statement, I love it. Available but non-leachable. When Hugh said that to me about 5 or 6 years ago, it just spun my brain out. But just write it down, available but non-leachable. It took me a while to really get a grasp of what he was saying, and that means that minerals are held with the humus or in the bodies of microbes. So you get a big rain, they're not going to run away. It's not going to leach. So
there's a white Audi out the front. I need to move that car because everyone's coming in.
we found the owner. So yes, everything's got minerals in it and an NPK value, but is it beneficial to the 3 aspects of the soil? And as we see what humus, and this is in your book, so it's going to increase and improve your microbiology, definitely improve the soil structure, because humus pushes soil particles apart. And Jerry Benetti, who's a great American presenter, said at Brisbane last year, he said, really what our soils lack is oxygen. And it's true. If we can get oxygen in there, then everything's going to kick along. So humus is going to push those soil particles apart and get some oxygen in there as well and hold your minerals. And guess what? We want our minerals that are in balance. We don't want them to leach and we don't want them to volatilise. So the trouble with conventional agriculture is we just keep putting more on of the same thing. And a lot of it is locking up, because we do total nutrient analysis, is it's locking up or volatilising, and we're losing it. So by keeping all of those minerals in balance, then we're going to grow healthier crops. So dead soil back to life. This was in a trial we did, so you can see this is what would be called a high hydrogen soil, not like Cam's got different soils to this, they're much blacker, and they're cracking soils, whereas we have high hydrogen around our district, but you can build this humus layer in your soils and bring them back to life again. So the formation of humus in the composting process is where much of the value is created, and we've spoken about this because it's the attributes of the humus to build aggregated soils and allow, what happens when we open up a soil? What will happen with water when we tip it on? Water does one of 3 things. It evaporates, it runs sideways, or it goes in. So if I pour water on this table, and we won't tell from what we did the other day, but the vase overleaked yesterday, but it just pool there, or it'll run away. But if it's a nice tilthy soil, where will it go? It'll go in. But also if it hits a hard pan, it will then go sideways as well. So we want to get the value of every drop of water that we can get, because water is going to be just becoming a valuable resource. Had someone the other day, he said, well, when you're in drought, you've got to worry about the flood, and when you're in flood, you've got to worry about the next drought. So I'm not sure what we're in at the minute, and I think it all keeps us wondering. Roots, this is my favourite part of the plant, and I've only got one slide on it. This is what I call the dreadlock roots. They're coated in the exudates. You've got the new growing root tip, which every 24 hours will be recovered with the microbes. The greatest microbial populations are in the root zone. 60% of your microbes live around here. If you were going to get fed cake and cookies all day, where would you live? In here. And that's why they sit around there, because they're getting fed from photosynthesis. There's a bit of a fallacy out there where you need certain fungi in the soil and bacteria, and it's more dominant. I believe that the plant will determine who lives around it by the exudates that it gives out. So much of what we know is, well, it's really what we don't know. We have to leave to nature. So it's really getting our soils into a condition where a lot of nature just takes over. And I find a lot of the scientific testing now is getting lost because they'll take X microbe and sit him outside and say, we'll test if your soil needs that microbe, but he doesn't have his friends who he's going to eat. Pretty dog-eat-dog world down there. And then they say, well, that microbe didn't work and we don't need that one. But it is so complex that, you know, it was only in 82 that we discovered glomalin or glomalin, however you like to say it, the sticky glues that hold soils apart. What else don't we know about our soils? All we know is if we get the soils in a natural, aerobic, tilthy condition with balanced minerals and microbes and humus, they will decide what is going to go on in the soils. And this is where I think our scientific minds prevent us from letting nature do its job, really. Trials. Oh, I'm on time. Okay, we did trials with Agritech, which is a local, well, they're now Australian-wide testing service, and we were researching and comparing 70 kgs of MAP with the Humus compost pellets, which we're developing, and BioTX, which is the humus compost made into a liquid. So it's like a compost tea, extracted rather than brewed. And they did all these little test sites on our son's property. What it showed is that the pellets, I was very, very happy with the 100 kgs of pellets for the liquid inject. I know they said MAP, but you put the liquid inject with MAP. So this is what we're saying. You could be conventional using 70 kgs of MAP, put a liquid microbial with it, and it boosts it even further. This was the 100 kgs and with the liquid and 15 kgs of MAP, and it lifted it up to about 7.1 tonnes to the hectare. So I believe we can just cut back our soluble fertilisers to absolute minimal. And as I said, this is a thing, if your inputs are high and your soil fertility is low, we don't tell you to do this overnight. You're going to be dropping it down little bit by little bit. You'll get confidence. Like we don't use fungicides, we don't use, what do you put on seed dressing? That's a fungicide, isn't it? Impact? I don't know. You know, we don't use fungal seed dressings. So as you get more confidence and your soils get better, you will drop out those things as your soils get healthier. So the healthier you can get it up front, the less you're going to need all the problems, and the nitrogen and the urea. I mean, I was only talking about the sides, down the other end.
Can I just add a comment to that? Try to say don't go cold turkey because that hurts big time.
That's what I said, don't do this overnight.
The soil's a drug addict, you need to wean it off.
Absolutely. So this is what I'm saying, if your soil inputs are high and your soil fertility is low, don't do that overnight. Do a little bit by little bit by little bit by little bit and just wean yourself off it. Because, yeah, a lot of people, well, no-tillers really go cold turkey in some ways. And they say, well, in 5 years, I'll get results. But really all they're doing is chemical agriculture. There's no microbiology in it at all. The liquid inject trial is, the liquid injecting, the ANOVA analysis of variance confirmed that it increased biomass, telecount, yield and proteins. This is the DPI trial. Now you have a piece of paper in your book that is that. I'd like you to open up to it. It's a threefold. And on one side you've got staple strength, and on the other side you've got these trials. So you'll need to flip it over. Yeah. And I don't have my copy here. This is the native pasture alternate fertiliser trial at Bynalong. There's been a lot of reporting on this trial, and I think it's been reported very poorly by the DPI, and they know it because I wrote to them. But if we're looking at improving soil structure first and foremost to make the rest of the soil functions work, you will see here, okay, this is it, and we haven't got into soil testing, so I don't want to go too far and too fast. So this was a compost mineral blend, this was single super. So the exchangeable calcium in this trial plot of ours was 29 at the beginning, 33 of the single super. In 1, 2, 3 applications of a compost mineral blend, we had the calcium at 63%. which was a 102% increase of calcium. Theirs was 23. The potassium, we actually were getting more imbalance. It went down 19, theirs went up 4. The magnesium, it was a very high mag soil, so it had 30% magnesium, which is way too high. It would be a tight soil. We reduced it back by 23%. Theirs actually increased. Now this is an interesting one. We decreased sodium as well. Theirs came down a little bit. The total carbon, we increased by 31%. Theirs was 17%. And the other one, if you look at pH, it went up 19, theirs was 8. The Coal P, even though we didn't add soluble phosphorus, we added soft rock phosphate and what was in the humus compost. Ours went up 14% and theirs was 19. But to me, if you look at the aluminium, ours went down 82%, theirs was 24. But they're reporting on this that ours didn't quite grow quite as much as theirs did. And it was more expensive, I will admit. But these changes are going to hang around a long time. And I don't think the soils around Yass and Gunning are suffering from a lack of single super. I think they're lacking a lot of humus myself. So that's something you can take home and have a look at. But as I say, they're just looking at growth. They're not looking at soil properties, which I think is a bit of a flawed trial.
The CEC's gone up there too, Rhonda.
Oh, yeah. Yeah, the CEC had gone up. Sorry, because where have we gone? Oh, down here. Does anyone know why our CEC would have gone up? 46 to 22. Who remembers clay holds the positive minerals? Everyone got that? What does humus hold? The positive and the negatives. So your storage capacity, which you really use CEC, cation exchange capacity, will grow. And as I said to Cam yesterday, we were having a chat, we've only got 4 CEC soils. Lucky Bugger's got 80 up there, so he's got a lot of positive minerals in his soil. There's no way my 4s will become 80, and there's no way Cam's 80 will become 4s. So whatever soil you're dealing with, we can make it better, but we're not ever going to do that big a change. We can grow soils, and we can make them better. So you don't have to sell your property and move on. This was a trial that Martin did on Knowledale, where we looked at 70 kgs of MAP against, and this was trial number 2, control 2 in the trial, against just total liquid inject. So we had the liquid inject against the MAP, and that was wheat, lupins and wheat. And we were doing total nutrient analysis to see we weren't mining the soil as well. So this was no granular fertiliser, that was pure liquid inject, and we mirrored the 70 kgs of MAP. It was very close, and financially you were much better off. This was a DE's DEC, you've heard of DEC, the Department of Environmental and Climate Change. They did trials on organic matter increases at 5 tonnes of hectare of compost. We wouldn't do that broadacre, we would only do that in horticulture and viticulture, but we increased it by 18.6% organic matter, which fixed, would have sequestered, when we say take out of the atmosphere, 22 tonnes of CO2, which would be a nice day at the office. And we were going to hold 56,000 litres more water per hectare. Who wants that at the minute when it's a bit dry? Hello? And you know what, if you've got open, tilthy soils, if you get too much water, what happens? It naturally will just drain away. It's not just going to pool at the bottom of the roots. So we talk about more droughts. There are more droughts going to occur with what's happening on the planet. Call it climate change or call it what you want. But we're going to get more unusual events. The hold this more water is going to ensure in your financial, in your outcomes, you're going to start putting more money in the bank. This was a CSIRO trial, and it's been run at the moment on the National Soil Research Program. This was the paddock we're going to look at today, called Old Sheepyard. And what it's showing is that this is your 3 pools of carbon, particulate, humus and recalcitrant. And in the top 10 centimetres, we're actually holding 53% of humus. So I rang Jeff Baldock the other day and I said, because I haven't released the findings yet, and he said the 53% is in the upper end of the trials done around Australia. Now that's comparing it even with high clay soils. Remember, we've got really light sandy soils. And he said if you divide the POC by the humus, it was 0.28, so you've got your labile carbon, which the bugs are about to eat. That's going to be eaten up by something. And you've got your humus, which is going to be around a long time. What it showed that our soil carbon is not vulnerable, so it's going to hang around. So, but if you had the other high labile carbon, which I see in a lot of soil tests, and low humus, that carbon is more vulnerable because it's going to be eaten by bugs and hopefully turned into humus. So holding, and this is where the whole carbon trading number is going to come from. They're going to be measuring the increases in humus. Unless you want to use biochar and put it into this pool, which is, to me, is a bit of a scary thing and we won't go there. But yeah, I was really happy. But the other thing is at depth, our humus actually increased at depth, which was, I don't know, I mean, interesting, really pleasing and interesting. Mulyan increased their baby leaf spinach, if anyone's into vegetable. The industry average is 1 kg, and they now yield 2.1 kgs per square metre of baby leaf spinach. Rocky East, for 10, 11, 12 years we haven't applied single super or urea onto Milgadara. So when you come on this afternoon and we are growing pastures such as this, lucerne chicory, arrowleaf clover, undersown canola. The canola would have had, and I've got to say this, we are still using a small amount of MAP at sowing time. Okay, so we haven't totally, we're in the middle, the biological, not conventional, not organic, we're in the middle. And this is where I think it's a really nice fit for most people, is to sit in that biological area. Nearly there. Wool staple strength has improved and it's controlled by protein nutrition. Now you've got another big sheet in there that we've done 11 years of staple strength data. Now the other interesting thing, don't forget that we started Wildland Living Soils in the first year of the longest drought in history. So all of these staple strength percentages and numbers have come right through a drought. So this year we had 100% of our wool staple strength above 30 newtons. So what do we get paid on for wool? Who's a wool grower here? Only 1, 2? What does everyone else grow? 3? Come on. Crops? Who's cropping then? Must be a lot of hands. Ah, cattle? Ah, that's why you've come to listen to Bill Hoffman. So we all know that productivity, getting our soils healthy, it's the productivity that we're going to drive. And what does productivity, increased productivity mean? It means profitability. And that's what we've got to become, is more profitable. A little bit of agri-organica, if I had 2 toolboxes, and I had the humus compost in this one, the other product I'd pop in there is a product called agri-organica. It's 3 food-grade microbes fermented in Japan with enzymes and minerals. $2.74 a hectare. You can add it with all your chemicals. And they've shown that if you add it to the soil, what it does to your native microbiology, and if you put chemicals, then you get all these new resistant pathogens. So if you're going out with a chemical, we put it in our liquid inject, we put it, if we do have to spray Roundup, we put it with our Roundup, liquid inject, foliar fertilizers, wherever we go out at $2.74 a hectare, 1 to 1,000 of agri-organica. This is, you'll see this crop today, canola, using the had a compost mineral blend on it and liquid inject, but only 15 kgs of nitrogen to grow that canola crop. So I want Hugh to comment on this when we're in the paddock. It appears that canola takes up boron more than wheat. So I want Hugh to comment on that as well. Green manuring, and this ends the presentation. I said to Bill after canola or lupins, I don't like the way that soils sit there and bake for 6 months before you go in and sow them. So I said, I really want to try some green manuring. So, you know, after the canola, this is after a canola stubble, bare soils do not fix carbon or feed microbes. Weeds grow on bare imbalanced soils. So some farmers are now having to spray 4 times between harvest and sowing just with glyphosate. And so we know what that's doing. That's now researched, and we know proven research of what those herbicides are doing to our soils. So to reduce the use of it is going to be a beneficial thing. So the purpose of the trial, I said to Bill, I really want to try some green manure. And he agreed, and we're going to look at this paddock today. We want to reduce weed burden and herbicide use. We wanted to increase organic matter, earthworms and beneficial microbes, increase the soil's available nitrogen and moisture retention, stabilise the soil to prevent erosion, provide a habitat, nectar and pollen for the beneficial insect, and improve water, root and air penetration in the soil. So if you had the choice, and particularly I think if we're going to get these Indian summers, you know, with all this rain, it's unusual and I don't know whether it's going to come forever or not. But if you had a bare dirt paddock or you could grow something on it that you're going to green manure in, I think you've got to start asking yourself, what is the better way to go? So the buckwheat was sown on January the 12th. It was speed tilled in on February the 23rd. So from that, it went to that. So we turned green material that was about 2 feet high, we turned it into the soil. So what were we feeding? When we feed green materials, we're feeding more of the bacteria. When we feed a brown manure, we're feeding more of the fungi. So, and also we wanted it green because we wanted to get the nitrogen from it. This was the Soils for Life visit with John Leggett. You'll notice there's low weed pressure there. We hadn't had to spray one spray of Roundup out. There were no compaction layers. It went right down. So this was in April, and there was no knockdown at that stage. So the soils had become tilthy. We'd increased, you know, you'd fed all the microbiology, and we hadn't had to put any sprays out. I think this is all positive still. There was earthworm presence, as you could see, and obviously this soil here, you know, you get down a little bit, we do get a little bit of that. We hit clay very low down, or not about that far down. So we're starting to open up the soil and feed these microbes. And we talked about mycelium. This was just on a piece of stubble. So would you say that's an active soil or a dead soil? Are you active or dead at the minute? Sorry. I'm going to feed you in a minute. Would you like to see that in your paddock? Okay. So we can then, so visually you can look at your soils, you can smell your soils, you can walk on your soils. Yes, a soil test is good, but really you've got to start using your own body and your own testing, you know, as a tester for all this soil structure and soil life as well. So this is the roots. This was on May the 23rd. Green manure crop, non-green manure crop. Now don't forget, lovely Dreadlock Roots, but don't forget this has still had liquid inject and it still had all the soft fertilizers, but look how much better that is. So improved soil structure. The leaf test, we did not put any foliars on this. So this is the leaf test, which is in the back of your book. So the findings so far, this is the Gregory wheat. It was only last week I took that photo. It's coming out into head. So it had one herbicide spray before sowing, had nil foliars, no urea and no insecticides. So I think so far, and it's only had 11 units of N and 11 units of P on it to grow this crop. So, you know, you can see for yourself. You can have a dig in there today and see what you think. Whether you think you're best to leave your soils bare, whether there's an economic value in that and spraying it all the time, or whether it's worth the extra to spend a little bit, put a green manure crop on and turn it in and feed your bugs. So this is just a bit of a trial that we're running, but I want to do all our canola country, or I've asked Bill, can we do it all again? James Fagan at Mulyan, he said, after applying the Wild Humus compost, we noticed a significant improvement in plant health, leaf vigour and root health in the vegetable crops, with most noticeable improvement in the baby leaf spinach. We noticed a significant improvement in baby leaf spinach, the yield was up and the leaf quality was much better, said Mr. Fagan. He's actually just bought his own turner, so he'll be making his own humus compost to put on his veggies. Michael Rickets, Michael has a bet each way. He does the wild humus compost, but he does conventional farming for the rest of what he does. He estimates that this wheat crop will go 8 tonnes dry land this year. And he's also worked out that it pays him every 3 years to spread a tonne of humus compost. He's worked it out in minerals, he's worked it out in yields, he's a figures man. So this fusion farming. He's pretty conventional, but he's still putting his good minerals in there and his humus in there, and he's really, really, really reaping the rewards. You'll probably win the crop comp with this locally, and hopefully maybe statewide. But you know what they call what we are doing? Unconventional. I love it when it's written up in the papers. So what we're doing is not biological, it's unconventional. So dare to be different and stand out from the crowd, challenge your belief systems and develop your own set of beliefs. So where to from here? Don't lose the momentum. Let's have a paddock muster in your area in 2013. So if you want us to come and have 4 or 5 people in your paddock, then just let us know. If we stand in your paddock, we observe your soils and look where do you want to go from here. So thank you very much. I know it was quick and fast and a lot in it, but thank you for listening.